timeline of the plant, and that timeline will be having difference in the term of anatomy, because there is a very characteristics different in the autumn wood and spring wood, and altogether it is commonly known as this annual ring. Now in the vascular bundle, again there is a presence of vascular resin canal. Resin canal is one of the prominent character. Generally resin is found in the yellow colored. When you compare this old as well as young one can see easily the difference in the characteristics of wood. In the young shoot, there is no difference of autumn wood and spring wood, because there is no annual ring formation, and that's why it is having only xylem element. While in the old one can see very well there is a difference in the annual rings, that means autumn wood and spring wood. Secondly, there is a difference in the phloem also. Primary phloem is having difference in both one. There is no difference in the cortex region. Cortex region is very common in both, whether it is young or old long shoot. The one more difference is young one is having epidermis, while old one is having cork. So cork is replaced by epidermis when it become elder one, and cork is giving lot of protection against microorganism, against lot many factors like cold or snowfall, even the rain, because that cork cells is having suberin, and that suberin is having a lot of resistance against microbes also, and generally this insects is not damaging the plant because of presence of cork region. Now after comparison we will go in detail of old long shoot at a cellular level, and this transverse section of wood, that is very important in terms of plant identification as well as in terms of evolution and lot many other aspects. Even economically, when the plant is used, lot many aspects are observed with the help of anatomy. The outermost layer of the old long shoot is the cuticle. In the general structure we cannot say cuticle, but when we go through the cellular level, there is a cuticle layer. That cuticle layer is a kind of waxy layer which is helping in the protection of water. It check or it stop the evaporation of water, and because it is waxy in nature, it is not permitting water to evaporate, and it is helping the plant in the terms of water loss. Next to epidermis there is cork region. Of course we discussed very well about cork, what is the exact importance of the cork, and there are few layers of the cork. And cork is having various part. Cork cambium is there, cork cells are there. So generally what we are finding in this section is the cork region, which is made up of dead tissue. It is tangentially elongated one, and it is giving lot of protection to the stem part or the shoot part. Following to the cork region, there is cortex, as we discussed earlier, and this cortex is forming the main ground tissue, or the what you can say outermost layer of the plant. This cortex is not the continuous one, it is having a lot of resin canal, and this next to cortex there is primary phloem and secondary phloem also. This primary and secondary phloem is forming the outer part of the cambium. Then one can see very well there is a very small layer, almost a single layer of the cambium, which is giving rise to xylem and phloem, and next to cambium there is wood. Here you can see very well there is a difference in the spring wood and autumn wood. One can find it very well, difference in terms of color. This wood is radiated by or interrupted by medullary rays, and resin canal is also present in the wood part also. And because of the presence of resin in the wood, this wood is not decaying or degrading very fast. Till the resin is present in the wood, it will remain for the long period. So many times this resin canal is resin is helping a lot in the wood preservation also. Next to wood, the secondary xylem, there is a primary xylem. A very small patch of primary xylem is there, which is generally not noticed when we take the section, but there is a very minor, very small part of the primary xylem is there, and followed to primary xylem there is pith region. In the pith region there are certain brown colored cells, that is tannin cells. So tannin is not found in the cortex region in thoroughly, or it is not found in the xylem part, but it is mainly found in the pith region. There is no much amount of tannin present in the entire wood when it become mature. Now for the three-dimensional study, or for the thorough study of wood, one has to go through the various part of the plant, the various kind of section. So here we will go through radial longitudinal section, that is RLS. When we take this section from the center, it is cutting from the radius part, and that's why it is known as RLS, that is radial longitudinal section. In the RLS of the wood we can find xylem rays, that is the one part, and this xylem rays are containing two different parts. The one is ray tracheids, and that ray tracheids one can see very well with the pit like structure, and the another part is ray parenchyma. Because this section is from the radius, so medullary rays are cut here, and this medullary rays are showing this ray tracheids and ray parenchyma. Such kind of ray tracheids and ray parenchyma will not be having similar structure when we take the tangential longitudinal section, that we will see later. The other part what we will see is the xylem parenchyma, and in the other part one can find a bordered pit. This bordered pit is a kind of connection between the another cells, and that bordered pit is surrounded by bar of cyano. That bar of cyano is a kind of structure which is there on the both side, upper side as well as on the lower side of the pit region, that is bar of cyano. When we take the TLS, that is transverse longitudinal section, this rays which we have seen earlier with the tracheids and ray parenchyma, ray tracheids and ray parenchyma, that will not be found here, but here one can find very well there is different kind of seriate of the xylem rays. Here you can see very well that there is a uniseriate xylem rays. This uniseriate is not showing much of the branches and not much depth or the width of the cells. This uniseriate xylem rays is a one kind, and second is the multiseriate xylem rays. Here one can see very well in not the only difference in the color, but even the multiseriate is having huge number of the xylem rays, so it is forming a bunch, and it is in the bunch, that's why it is multiseriate, while uniseriate one is having only the single layer of the rays, and the junction of two rays is forming a special structure which is known as torus. The torus is alternating to the various uniseriate rays. So in the transverse section, transverse longitudinal section, TLS of wood, one can find multiseriate and uniseriate xylem rays and torus. Again, the important one, that is the RLS, the comparison between RLS. In the RLS one can see the bordered pit and bar of cyano, as well as one can see the ray tracheids and ray parenchyma, while in case of TLS of the wood one can see multiseriate xylem rays and torus. So this is the difference between TLS and RLS of the wood in case of Pinus. Now when we take the section of dwarf shoot, which is very small and with limited growth, we can see entirely different structure, and that structure is quite different in comparison to the long shoot, but there is certain characters which is remaining same. There is no major difference in terms of anatomy. Here in case of the dwarf shoot, the outermost layer is epidermis. Because it is not having the long life, because it is having a limited growth, it is having limited life, it is not forming cork here, but here the outermost layer is epidermis. This epidermis is surrounded by scaly leaves. Now this scaly leaves is not found in the case of root, of course, in the case of old stem also, in the case of long shoot also. Long shoot is not having surrounding of the foliar leaves, but here in the case of transverse section of dwarf shoot, one can see very well that epidermis is surrounded by scaly leaves. The major part of dwarf shoot is made up of cortex, and this cortex is containing again lot of tannin cells. This dwarf shoot is more prone to the animal disturbance, and that's why there is a presence of tannin cell which is helping in the protection from the animals. Here again one can see the presence of resin canal in huge amount. It is forming a circular ring like structure, and this resin canal is found in cortex region very well. There is no clear cut development of endoderm. There is no clear cut development of pericycle. Endoderm and pericycle is not found here. Directly it is forming phloem. Directly phloem region is there, and phloem is surrounded followed by xylem. In between the phloem and xylem there is a cambium which cannot be easily distinguishable here in the case of general structures, right? And the central part is made up of pith region. Of course, pith is one of the common characteristics. This pith region is also containing presence of tannin cells. Now when we go in detail of the transverse section of dwarf shoot, we can see epidermis, which is made up of single layer. It is almost having the square shape or tangentially elongated cells. There is no radial elongation, but here the tangential elongation is found in the case of epidermis. Outside the epidermis there is one can see the presence of scaly leaves. Of course it is not attached, it is bit away from the epidermis. Then there is a huge amount of cortex, a few layer of cortex, and cortex is containing resin canal, and as well as it is containing tannin cells also. Now here the primary phloem is very small, and this primary phloem is crushed, and sometimes it is found in the form of patch only, which remain generally unnoticed, because one can see the presence of secondary phloem. Secondary phloem is continuous one. It is made up of few layer, that cells are very prominent, one can see very well. In comparison to secondary phloem, this primary phloem is containing very small patch and very small amount. That's why primary phloem is not much distinguishable in the case of dwarf shoot. Followed to secondary phloem, there is of course presence of cambium, and cambium is followed by wood. Here you can see very well there is no much difference in the wood, because it is dwarf shoot. Dwarf shoot is not having long life, it is having limited growth, and that's why there is no presence of autumn wood and spring wood or annual rings. There is no formation of annual rings. There is no formation, or there is no specificity of autumn wood and spring wood in case of dwarf shoot, because dwarf shoot is having limited life. But this wood is also showing two different kind of medullary rays. One is the primary medullary rays. This primary medullary rays is starting from phloem part, and it is radiating toward the center, while there are presence of secondary medullary rays also. This secondary medullary rays are starting from the cambium part, so it is found only and only in wood region. It is not found in the phloem part. But primary medullary rays is starting from the phloem, from the primary phloem, and it is continuous up to the wood region, it is continuous up to the secondary layer. So this primary medullary rays and secondary medullary rays is found only and only here in the dwarf shoot. It is not found in the long shoot section, right? Then toward the center, when we move, we will find presence of primary xylem, and this primary xylem is made up of protoxylem and metaxylem both. And this protoxylem is found toward the center and metaxylem is toward the periphery. And this primary xylem is showing lot of difference from the secondary xylem, that is the wood part. In the primary xylem part, there is no presence of medullary rays, and the centermost part is made up of pith, and this pith region is also containing tannin cells. So in the dwarf shoot, tannin cells are found in cortex as well as in pith region, and the characteristics part of this dwarf shoot, transverse section of dwarf shoot, is there is a medullary rays which is of two different kind, that is primary as well as secondary. When we take the uppermost region of the dwarf shoot, from where the foliar leaves are radiating, from where the foliar leaves is growing, we can see the structure according to the number of foliage leaves. If there is two foliage leaves, one can see the tri-radiating part, otherwise semicircular part. Here we can see the triangular part. In the case of dwarf shoot, we will go briefly with the dwarf shoot. The outermost layer is epidermis, the same which is there in the case of normal structure. Even in the base, the same thing is found. Then epidermis is followed by cortex. Huge amount of cortex is there. Presence of resin canal is again here, and this resin canal is continuous up to the leaf part. Whatever the number of resin canal is here, the similar number of resin canal will be there in the leaf also. And the centralmost part is the vascular bundle, and this vascular bundle is a direct supply toward the leaf, that is of acicular in the shape, that leaf needle or Pinus needle, what we are commonly knowing. When we go through the transverse section of leaf needle, we will find the outermost layer of epidermis, and this epidermis is made up of a single layer, and that is not continuous. One can see very well that it is not continuous in nature. It is interrupted by presence of stomata. Now because